All right, in the second half of this unit, we are going to look at some chemical energy and also talk about cellular respiration. This chemical energy is really important because it is how we actually get the energy that we need in order to power our life processes. So let's talk a little bit about that chemical energy because we did already mention it when we talked about photosynthesis. But the energy in chemicals, including glucose, are stored in the bonds between the atoms. Now, the energy inside of the cell, though, is stored in the form of ATP. And ATP's full name is adenosine triphosphate. So it's made up of, a base molecule that has 3 phosphates hanging off of it. So how come we need to use this ATP, and why can't we just use glucose directly? Well, there's a couple of reasons. Number one, ATP is reusable, so we can use it over and over and over again, whereas glucose, it's not that easy. We can't make glucose ourselves as humans because we don't undergo photosynthesis, but both plants and animals actually use ATP. ATP is also very small. It cannot what we call polymerize. In other words, it can't form really, really, really big molecules, and so it can be inside of every cell. Sugar can. Glucose can actually form a big long chain if you stick a bunch of glucoses together and make starch. And the last reason is the bonds in ATP actually contain more energy. They're what we call high energy bonds. So if we take a look at how this actually happens, we start off with ATP. So you can see here we've got our adenosine and then 3 phosphates that are hanging off. And these little lines in between the phosphates are the bonds. This is our stored energy, and that's where it's kept. So what we do is we turn it into ADP plus a P, so we break this bond here. So what ends up happening is the ATP ends up looking like this, and notice that I only have 2 phosphates on there. So I get 1 loose phosphate that's kind of popped off, and then that bond, when we break it, releases energy. So let's take a look here at our ATP handout. It says ATP is the basic source of energy of all cells. Energy is stored by cells when ADP is converted into ATP. Energy is released when ATP loses a phosphate and becomes ADP. So we need to label which one is energy storing and which one is energy releasing. So if we look at the top one, we're starting with ADP and phosphate, and we're making ATP. So what we are doing is we are storing our energy because we're forming this new bond right here, which has the energy in it. Then we have ATP, so 3 phosphates, and we end up with ADP plus a phosphate. So we have now broken this bond, and it is energy releasing. Okay. So let's answer the questions at the bottom. How many phosphate groups are in 1 molecule of ATP? We have 3 of them. How many phosphate groups are in 1 molecule of ADP? Di is 2. What are the 3 parts of an ATP molecule? Now, we didn't go into this as in depth, but it is an adenine, and that's the same adenine that you find in DNA, a sugar, and then 3 phosphates. So, if we think back now to photosynthesis that we talked about before, we need that glucose that we made at photosynthesis in order to produce ATP. Basically what we're doing is we're taking the energy in the glucose bonds and we're shifting it to the usable form of cell energy in ATP. So we know that photosynthesis produces glucose, but we're going to have to change that from that form of chemical energy into cell energy, which is its usable form. So what are some things that actually affect our photosynthesis reactions? The light intensity, how much light we get, which actually makes sense. If we don't have light, we can't run those light reactions in the thylakoid membranes. The amount of carbon dioxide, again, we need that carbon dioxide to make the glucose. And the last thing that maybe isn't as obvious is the temperature. That's one of the reasons why plants and trees lose their leaves. over the winter is because the temperature actually drops too low for them to undergo photosynthesis. So once we have this glucose, how are we going to shift it into ATP? Well, that process is cellular respiration. It changes food energy, our glucose energy, into usable cell energy in the form of ATP. Now, the differences are between eukaryotes and prokaryotes with this. This takes place in both plants and animals. This is something that plants have to do as well. They don't just do photosynthesis. In eukaryotes, this takes place in the cytoplasm and the mitochondria. So as long as you have a nucleus, you can do it in both places. In prokaryotes without a nucleus, they don't have mitochondria, so they can only have this take place in the cytoplasm. So let's talk about these mitochondria. Mitochondria are small, I almost say they look like a little bit like a fat hot dog shaped organelles. And like I said, they are found in both plant and animal cells. So this process takes place whether you are a producer or a consumer. Both types of animal or organisms have to have these or have to undergo this process. There's an outer membrane that's smooth, and then the inner membrane is folded. And these folds have names. They're called cristae. Basically, they are there to increase the surface area for the different chemical reactions that we have taking place. So this is kind of what they look like. All right, so you've got the smooth outer one, and then you've got the folded inner one, with those folds being called cristae. So let's take a look here at the diagram that's on your handout, page 84. So in plant and animal cells, the final stages of cellular respiration take place in mitochondria. A mitochondrion has two membranes. The inner membrane is folded up and the outer inside of the outer membrane. The space between the inner and outer membranes is called the intermembrane space. The space inside the inner membrane is called the matrix. So we need to do some labeling. So we're going to start over here on the left. That one is pointing to our outer membrane. And then up top, we're in between the outer membrane and the other thing. So this is our intermembrane space. We also then have the folded thing, which is called the inner membrane. And then inside of the inner membrane is the matrix. So we also have a question with this. In which membrane is the electron transport chain located? Now again, we haven't talked about this, but what we will learn is it is along the inner membrane. So let's get into the process of cellular respiration here. The first step in the process is called glycolysis. So this is the first set of reactions for respiration, and it occurs in the cytoplasm. Basically, what's going to happen with glycolysis is that glucose literally gets broken in half. Remember, it is a 6-carbon sugar, and so when I break it in half, I get 2 3-carbon sugars. It's just like snapping a pencil or snapping a carrot. When we do this, we get 2 ATP, and we say this is a net gain because we actually have to use a little bit of ATP to get things started, but we end up with 2 extra ones on the other side. So basically what happens is we take glucose and 2 ATP, and then this is NAD, it's another one of those electron energy carriers, or high energy electron carriers. And then what we get out of this is pyruvic acid, which is what happens when you split glucose in half. But notice we get 4 ATP, so 4 minus 2 that we put in leaves us with a net gain of 2. And now we've actually captured some energy on our electron, high energy electron carrier in NADH. So again, glucose is 6 carbons, pyruvic acid is 3 carbons. NAD plus is that high energy electron acceptor. NADH is one that is basically an NAD plus that's accepted one of those high energy electrons, and we're going to use it later in the electron transport chain. Now, what happens after glycolysis depends on the type of the cell and whether or not oxygen is present. So this is what we've done so far. We've taken glucose and we've gone through the process of glycolysis. Out of that, we get 2 ATP and 2 NADHs. So after glycolysis, it's going to depend on if oxygen is present or if oxygen is absent. If oxygen is absent, we undergo fermentation, which is anaerobic. So anytime we have oxygen absent, we refer to it as anaerobic. If oxygen is present, then we move into aerobic respiration. We're going to take a look first here at fermentation because it's a pretty simple process. Anaerobic respiration, like I said, is also called fermentation, and it does not require any oxygen. This occurs in the cytoplasm. Now, the thing with fermentation is we don't get any more ATP produced at all. We use the NADH in the fermentation reaction, and this process mostly happens in prokaryotes, but it can happen in eukaryotes where oxygen is scarce. So if we do like an anaerobic style workout, your muscle cells will actually undergo this process rather than the regular aerobic respiration that your cells normally do. So with anaerobic respiration, we start with that pyruvic acid, and we actually have two different types of fermentation that takes place. In the first type, we get alcohol and carbon dioxide produced. In the second one, we get lactic acid. So these are different processes because we have different products and they have different names. This is alcohol fermentation that takes place in yeast. So if you think of things like making beer or wine, this is where we get the alcohol and the bubbles that we get in things like beer or even champagne. This also takes place in things like bread making. Now the alcohol, when we cook our bread, the dough, it actually evaporates off, but the carbon dioxide creates the bubbles in the bread that allows the bread to rise. When we make lactic acid, that's lactic acid fermentation, and I'm sure many of you have heard of that in your muscle cells. So we need to move over here to our glycolysis and fermentation handout. And at the top it says glycolysis uses ATP to break a molecule of glucose in half, producing pyruvic acid. When oxygen is not present, glycolysis is followed by fermentation. Fermentation enables cells to produce energy in the absence of oxygen. So we're going to follow the prompts to identify the important parts of glycolysis. So we're going to start off by coloring the carbon atoms blue. So we've got blue carbon atoms up here. We've got blue carbon atoms here, 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 and here, here, and here. You guys can color yours more neatly than I am coloring here. It says to circle the place where ATP is formed. So if you look here, we're going from ADP and we end with ATP. And it says mark an X on the place where ATP is used. Well, up here, we start with ATP and we end up with ADP. So that is where ATP is used. So again, 4 minus 2 gives us a total of 2. All right, how many carbons are in one molecule of glucose? Well, we know that that is 6. And what is the product of glycolysis? That is pyruvic acid. So again, this is a pretty simple process with fermentation. It just does glycolysis and fermentation. Where we actually spend most of our time, though, is with the aerobic respiration. Aerobic respiration is the one that requires oxygen. And we have two major parts to it, the Krebs cycle and the electron transport chain. Now, prokaryotes can have this take place every so often using the cell membrane in the cytoplasm, but the majority of organisms that undergo aerobic respiration use the mitochondria. So, the first step in aerobic respiration is called the Krebs cycle. The Krebs cycle produces carbon dioxide, hydrogen atoms, and ATP. Now this occurs in the matrix, so the very inner part of the mitochondria, or in the cytoplasm if you're talking a prokaryote. Now the Krebs cycle is a turning cycle, just like the Calvin cycle. It goes round and round and round. So for each turn of the Krebs cycle, we get for each glucose molecule, which is actually 2 turns of the Krebs cycle, we get 6 NADHs, 2 FADH2s. Now this is another electron carrier. It's just a different name. And we get 2 ATP. So, but we're not completely done yet. So we get 2 more ATP just through the Krebs cycle from a molecule of glucose. We started off getting 2 from glycolysis, and now we actually get 2 more from the Krebs cycle. Now, this NADH, not just the NADH from here, but also from the glycolysis, as well as the FADH2, they go to something called the electron transport chain and actually help us to create even more ATP. So let's take a look here quickly, though, at the Krebs cycle so that we can take a look at how we get this. So the Krebs cycle. If oxygen is present, the pyruvic acid formed during glycolysis moves into the Krebs cycle. The Krebs cycle converts pyruvic acid into carbon dioxide. As carbon dioxide is formed, high-energy electrons are accepted by NAD+ and FAD. This results in the formation of NADH and FADH2. NADH and FADH2 will be used later to produce ATP. So it says, follow the prompts to identify the important parts of the Krebs cycle. So it wants you to color all of the carbon atoms blue. Okay, so we've got blue here, we've got blue here, there's also blue there, blue, 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 another blue with carbon dioxide kicking off again, and blue. Okay, it says circle the electron carriers in green. So the electron carriers here are NADH and our FADH2, another NADH, and another NADH. And it says circle the ATP in orange. Okay, so ATP is produced right there. Okay. So, question here. Which of the following is formed during the Krebs cycle? We actually form FADH2. We use the pyruvic acid. We do not form it. So that is the first part of aerobic cellular respiration. The second part is the electron transport chain. So this is the second phase of aerobic respiration. If I'm a prokaryote, we can use the cell membrane on the inner lining of it to do this. But in eukaryotes, we use the lining of the inner mitochondrial membrane, so the inside part that's right next to the matrix. This is going to produce ATP from NADH and FADH2. So what happens here, and this is kind of a complicated process that we're trying to simplify so you guys can understand. Basically, the NADH and the FADH2 supply high-energy electrons, and these electrons get passed along the membrane in the electron transport chain. As they move along, they lose energy. What this does is it sets up a hydrogen ion concentration gradient along the membrane. So we have a high concentration on the outside and a low concentration on the inside. And what that allows us to do is to create ATP by a process called chemiosmosis. And we're going to detail that more here, but remember this also takes place in photosynthesis when we produce some ATP in the light-dependent reactions for use in the Calvin cycle. But let's review the different things that are happening here in the electron transport chain. So the electron transport chain uses the high-energy electrons produced by the Krebs cycle to move hydrogen ions from one side of the inner membrane to the other. So we have to start off here first doing some labeling. So we need to label electrons. And the little e with the negative sign is an electron. Now, we also have to label the hydrogen ions. So the H with the plus is a hydrogen ion. And then the last part is the inner membrane, and that's what is bracketed here, is the inner membrane. Okay. So let's answer the questions now. Where in the mitochondrion does the electron transport chain take place? That is along the inner membrane. So what do we do with the high energy electrons from the Krebs cycle? We're going to convert ADP into ATP. And that's going to happen through the process of chemiosmosis that I'm going to explain to you now. So chemiosmosis is how we produce ATP. So this happens in photosynthesis because we need some energy in the Calvin cycle, and we also need to create it using the electron transport chain in cellular respiration. All right, so in photosynthesis, when we split our water, we get H+, and so now we have some on the inside of the thylakoid. Now, that's basically an energized electron. So the energized electron provides energy to pump in even more H+. So we get that from NADPH in photosynthesis or NADH in cellular respiration. So this is what it kind of looks like. So it sets up a concentration gradient. So you guys can draw this. This is your membrane, okay, and we have H+. So we have a low concentration on the outside and a high concentration on the inside of the membrane. And there's this little molecule here called ATP synthase, and it spins like a top. And as it spins, it pulls hydrogens from the inside to the outside, and through that process, it actually creates ATP. Now, I'm referring to this in terms of photosynthesis because your handout is based on photosynthesis. When we get to the handout, we'll actually also relate it to cellular respiration. So, as the H+ moves out of the thylakoid into the stroma, ATP synthase captures the potential energy of the gradient. Remember, potential energy is a form of energy. In the mitochondria, it moves out of the matrix and into the cristae, those folds of the inner membrane. Every time we move an H+, we get an ATP. And what it is doing, it is changing potential energy into chemical energy. So let's take a look at the process of chemiosmosis. Now, I'm not going to read the stuff up top because it relates to photosynthesis. But what I want to do is I also want to draw the parallels here with cellular respiration, because the labels here on the side, on the left-hand side, are set up for photosynthesis. So the inner thylakoid space is on, basically on the inside of, if we think of a granum, we're talking in here, okay? This is also where the matrix is in our mitochondria. And so then we have the thylakoid membrane, or this would be the inner membrane of the mitochondria. And then the stroma would be the intermembrane space in the mitochondria. Because remember, this takes place in both processes. Okay? So color the arrow that shows how ATP synthase spins. Okay? Right here. This one right here. This is the spinning. And like I said, it spins like a top. And then it says to draw in the formation of ATP from ADP. So as it spins, we take ADP and a phosphate, and we make ATP. That's what's happening. Okay, question with that one. Where does the formation of ATP take place? In the stroma, or if we're talking mitochondria, in the intermembrane space. So why do we have this all taking place? Well, remember, the electron transport chain is going to create the ATP, but it's also going to produce these electrons. So the electrons need a final place to actually go at the end of the chain. If there isn't a place for it to go, the chain cannot continue. So the final acceptor in this excuse me, is going to be oxygen. And this is how it works. The oxygen takes 4 electrons that have gone through the electron transport chain and 2 of the hydrogen ions, and it makes water. So the electrons are from the electron transport chain, and the H pluses are coming off of the NADH and FADH2 as part of chemiosmosis. So, how much ATP do we actually get from the electron transport chain? Well, each NADH makes actually 3 ATP, and each FADH2 makes 2 ATP. So let's take a look at how many total ATP we get by going through the process of aerobic respiration. So remember, we start back with 1 glucose and we go through glycolysis. Glycolysis produces 2 ATP and 2 NADHs. We just said each NADH makes 3 ATP, so I get 6 more ATP through the electron transport chain with that. Then glycolysis makes pyruvic acid. Okay, pyruvic acid through the process of the Krebs cycle, gets 2 more NADHs, which makes 6 more ATP. It turns into something called acetyl-CoA. We don't need to worry about that, but we get 2 turns of the Krebs cycle, and this directly produces 2 ATP, 6 NADHs, and 2 FADH2s. The 6 NADHs gives us 18, and the 2 FADH2 gives us 4. So when we total that up, we get a grand total of 38 ATP. Now you compare that with 2 ATP that we get from fermentation, and this is why aerobic respiration is so much preferred by cells over fermentation. So let's take a look at the cellular respiration reaction and then do a couple of things to kind of finish up. So we have 6 oxygen plus glucose. Remember that C6H12O6 is our glucose. And we get oxygen and water plus 38 ATP, which is our energy. Okay, so now we need to switch over to the last few handouts that we have. The first one is our overview of cellular respiration. So kind of like what we did with photosynthesis, we're actually going to kind of give you a visual here of the process. So this is the process, and we're going to take a look at what 1, 2, 3, 4, and 5 are. So glucose inserts into number 1, which makes pyruvic acid and electrons carried in NADH. So that first process is glycolysis. Off of glycolysis, we get number 2, which is again ATP. And the pyruvic acid from the glycolysis feeds into number 3, which is the Krebs cycle, and you can see it turns around and around. Number 4 is where this takes place. The Krebs cycle is in the mitochondrion. And then the electrons from both glycolysis and the Krebs cycle head over to number 5, which is the electron transport chain. So, where does glycolysis take place? In the cytoplasm. And where do the Krebs cycle and the electron transport chain take place? In the mitochondrion. So the last 2 things to kind of finish up with cellular respiration, we have a compare and contrast table here that we're going to do on the next slide. So the first one is the function of glycolysis. We're going to get quick production of ATP. We need some quick energy as well as high energy electrons. Fermentation is allowing us to release that energy without without oxygen being present. Sorry, that's a typo. That should be oxygen, not CO2, so just take that out there. Cellular respiration is slow production of ATP, but it's with oxygen and it's more efficient. The reactants for glycolysis are glucose and ATP, and they are the same for fermentation. Notice for cellular respiration we have pyruvic acid and high energy electrons. The products of glycolysis are 2 ATP and 2 NADHs. Fermentation, we get NAD+, alcohol, carbon dioxide, and lactic acid. Notice it does not say any ATP there. For cellular respiration, we get 38 ATP, 6 oxygen, and 6 waters. Advantages of fermentation, we can release energy without oxygen. Advantages of cellular respiration, we get more ATP and it's a longer term production. Disadvantages, for glycolysis, it quickly stops, and for cellular respiration, it is slower than fermentation. And the last one is a compare-contrast specifically with photosynthesis and cellular respiration. So the function here, so cellular respiration and photosynthesis can actually be thought of as opposite processes. Energy flows in opposite directions in the 2 processes. So we need to complete the table using the words in the word bank. So if photosynthesis is energy capture, then cellular respiration is going to be the opposite, which is energy release. Location. Photosynthesis takes place in the chloroplast, whereas cellular respiration takes place in the mitochondria. The reactants of photosynthesis are water and carbon dioxide. For cellular respiration, it's glucose and oxygen. Notice here, the reactants here are the products here. So the products of cellular respiration, we produce water, and we also produce carbon dioxide. That's one of the main reasons why they are opposite processes. The reactants in one are the products of the other, and so these guys just keep cycling around. We make water to oxygen, it goes over to here, and then cycles around. And the last one, last set of questions. Which process releases energy of the cell? That would be cellular respiration. For which reaction is 6CO2 plus 6H2O yielding glucose, C6H12O6 and 6O2? Well, I have carbon dioxide and water. Those are my reactants. That is photosynthesis. How do the products of photosynthesis compare to the reactants of cellular respiration? They are the same.